Thank you for joining the nSolver video tutorial series. This video covers creating advanced analysis visualizations from nSolver data. In this video, we'll cover preparing your data in nSolver to make your advanced analysis more streamlined. We'll go through the steps to initiate an advanced analysis in nSolver, and we'll also cover the differences between a quick and custom analysis. We'll go over the following visualizations: overview plots, normalization, differential expression, or DE, gene set analysis, or GSA, path view, and SNV, and the available customizing options for each of these. Cell type profiling, probe descriptive, related analytes, and the fusion modules are not covered in this video. Open nSolver. You should have already imported your data and created an experiment. If you missed these steps, see our Import and Quick Start for 3D Data video in this series. For this example, we'll use a practice dataset that is included with your nSolver download, the 3D Bio dataset. This data contains 3 biological replicates from 2 different melanoma cell lines. The cell line SKMEL28 has a known mutation in both copies of the BRAF gene. The cell line SKMEL2 has 2 normal copies of the BRAF gene, as well as a known mutation in the NRAS gene. Both cell lines were treated for 8 hours with either DMSO as the vehicle, or vimurafenib, which is a specific inhibitor of the mutant BRAF protein. As you create your experiment in nSolver, keep the following in mind to prepare your data for advanced analysis. You may wish to assign shortened labels that uniquely identify each of the RCC files, including the SNV references if you have any, using the description column. This will simplify downstream visualizations. In addition, create annotations that will be informative to you in your analysis. For this example, we'll use 2 annotation categories: treatment and BRAF genotype. Under treatment, we'll assign DMSO or VEM according to what is documented in the sample names. Under BRAF genotype, we'll assign wild-type to the SKMEL2 samples and mutant to the SKMEL28 samples. You can leave background, normalization, and ratio creation off, since advanced analysis has its own methods of addressing low counts and normalization, and will moreover be selecting raw data for our analysis in this example. Your experiment should be visible on the Experiments tab. Expand the navigation tree by clicking the plus sign to see all levels of data. Select the raw data under the experiment name, and then select all samples in the central table. Select the Advanced Analysis button. This will launch Advanced Analysis. Choose a name for your analysis, and select nCounter Advanced Analysis 2.0 for analysis type. Browse to choose where the output visualization files should be saved. Select Next. Select a unique identifier. This field will be used to label samples in the resulting plots. If preferred, use your modified labels that you entered in the description column when you were preparing your data in nSolver. If you have SNV data, make sure that your SNV references have been labeled with the same sample identifier. In the Use for Analysis column, select the variable or variables to use as covariates for analysis. There are several to choose from. For this example, we'll use the annotations we created earlier: treatment and BRAF genotype. Select Next. In a quick analysis, the analysis is performed with default parameters, and the core modules are pre-selected: overview, normalization, differential expression, GSA, and path view. Only a single covariate can be used in a quick analysis. In a custom analysis, multiple menu tabs will appear to the left of the screen, allowing you to customize parameters. Probe annotations are required for GSA, path view, cell type profiling, and pathway scoring. Default probe annotations are preloaded for NanoString commercial code sets. If using a custom code set, however, you'll need to generate your own probe annotation file and import it by clicking the Here link at the bottom of this window. Refer to the Advanced Analysis User Manual for more details. You can also contact NanoString support for assistance on probe annotation files. In addition to the core modules, you'll have access to related analytes, probe descriptive, cell type profiling, and pathway scoring. You can select or deselect these to customize your analysis. You may select multiple covariates in a custom analysis. For this example, we'll select Custom Analysis. Select the General Options tab. For the purposes of this example, deselect any modules other than Overview, Normalization, and Differential Expression. We'll focus on these to get a general overview of our data and the samples and genes that are differentially expressed in it. On the Normalization tab, you can customize the normalization settings. For this example, leave all the defaults. On the Differential Expression tab, move Treatment and BRAF genotype from the Available Annotations field to the Selected Predictors field using the green arrow button. Leave the optimal settings and p-value adjustment method as defaults. Leave the Run GSA and Display Results using PathView boxes selected. These plots will provide more detail and context to the differential expression results. We can change the number of top pathways to display from 20 to 10 to speed up processing. Leave the Color Plots By and p-value threshold settings as defaults. Select Finish. You'll ultimately be returned to the nSolver dashboard. Expand the navigation tree of your experiment on the Experiments tab and highlight the analysis data level. Highlight the analysis you just ran in the central table and select the Analysis Data button. Your analysis will start to load in an HTML window. You may need to allow blocked content. Allow up to 1 hour, potentially, if running Advanced Analysis 2.0 for the very first time, as the initial downloading of the R libraries is time-consuming. You'll need an internet connection and permissive firewall settings for this step. The overview heat maps cluster data with similar expression patterns. Colored bars along the top of each heat map designate SNV and fusion variant status, covariates, and QC flags. The colored bar along the left side provides information on probes that will be dropped from analysis due to low signal. In the raw data heat map on the left and the normalized data heat map on the right, we can see roughly how the data is clustering and if that coincides with any of the variants or covariates. In this example, there are no QC flags, hence the solid colored bar. We can see that one sample, 2-DMSO R3, appears to be slightly different from the others. This sample does not deviate a great deal, so has no cause for concern, but it serves as an example of the type of pattern you can look for to identify outliers in your data. We won't draw any additional conclusions from these plots since this module is intended to be used as a QC tool and a way to get a general impression of your data. In the Principal Component Analysis, PCA, a clear separation of BRAF genotype data points can be seen in the PC1 versus PC2 results, meaning that changes in this variable cause clear, consistent changes in the data. This is reinforced by the p-value histograms under the Other QC tab, which shows a clear left-weighted plot for the BRAF genotype samples, meaning there are a number of p-values in the significant range, close to 0. The treatment p-values are more evenly distributed, indicating that relatively few genes appear to be differentially expressed between treatment and control samples. The scatter plot on this tab displays the housekeeping genes in color. Their placement at the bottom of the plot indicates that they are stable and require little further attention. Let's select the normalization module next. Looking at the mRNA data, we can see the effect of the GE-norm algorithm's removal of each candidate reference gene on the variability of the normalized data. This information is then used to rank them. We can also see the mean square error, MSE, of each sample plotted against its normalization factor in the normalization summary plot. Samples that are outliers are named on the plot. We can see that 2-DMSO R3, again, appears as a bit of an outlier. Under the Protein tab, we can see similar plots referencing proteins that were unselected for normalization and the MSE versus normalization factor for each sample in the normalization summary. We can check protein controls on the protein expression background plot. Select the Differential Expression tab. The volcano plot for the covariate BRAF genotype depicts the differential expression of genes in mutant samples relative to the wild-type samples. It shows multiple p-value significance level thresholds. Only probes with p-values in the significant range are colored and named. Viewing this plot under the Treatment tab shows a colorless plot with no p-value thresholds, indicating treatment did not result in significant gene expression changes. Select the GSA tab. Gene Set Analysis, or GSA, shows us the variation in global significance scores among the gene sets for each covariate. BRAF genotype is associated with more variable results among the gene sets than treatment. We can see from the directed global significance scores plot that the PI3K-AKT pathway gene set has the highest score in the BRAF genotype category. Selecting PI3K-AKT from the list on the left results in a familiar volcano plot. This is the differential expression volcano plot, overlaid with colored points, which reflect the probes in this selected gene set. We can see that there are a number of probes from this gene set with significant results. Select the PathView tab next. As a next step in the GSA analysis, we can view the pathways that include our gene set or gene sets of interest in the PathView module. Here we select the PI3K-AKT pathway to see where our genes of interest lie in this particular pathway. Colored boxes show the specific elements of this pathway that were differentially expressed and whether they are up or down regulated in our data. If we decided to later run the probe descriptive module, these significantly affected genes would be good candidate genes for inclusion in that module. If you have SNV data, the SNV tab will appear. The SNV call summary gives a clear depiction of the SNV calls made in this data. Results are as expected. SKMEL28 samples all exhibited variant calls in the BRAF gene, while SKMEL2 samples all exhibited variant calls in the NRAS gene. The By Samples tab allows you to view a plot of each individual sample and the data for each individual gene tested for that sample. For more information on SNV analysis, see the All About SNV Analysis guide. Returning to the nSolver dashboard, we can see all of the analyses we created have been automatically saved under the analysis level. You can also find a folder of automatically saved images in the location indicated when you first created your analysis. This concludes the nSolver video tutorial series lesson, Advanced Analysis Visualizations. You can always contact NanoString support and check out our other video training curriculum online. Thank you.